Okay, thank you. Let me know if it's not loud enough, and I'll try and adjust at this end too. But thank you very much to the organisers for inviting me to present to you today. I apologise for not being there in person. We are currently having site reviews by our funding agencies, and I think everybody in the audience will appreciate how much we need to keep those people happy. So that's why I'm in Cork today and not in Oxford with you. But I'm going to talk to you today about some of the work that we've been doing targeting Listeria monocytogenes with phage-derived proteins. Okay, why is it moving? Okay, here we go. Well, let's talk about Listeria monocytogenes. And these are some data from 2021 from the European CDC, where they provide information on all of the different foodborne pathogens in the EU. And you can see here from these numbers, the numbers of listeriosis cases in the EU in 2021. So about 2,000 cases per annum, which is not a lot really when you compare it to the likes of salmonellosis or campylobacterosis. We're talking about, you know, maybe 500,000 cases per year. And it's often a question actually in when we submit grant proposals to work on Listeria monocytogenes. People often say, you know, why don't you work on campylobacter? Why don't you work on salmonellosis? But one of the reasons that we focus on this particular organism are these numbers here. So if you look at 2,000 cases, almost half of those people end up in hospital. And the case fatality rate in 2021 was 9%. It can be much higher than that. It can be up to 30% in some years. So this is a serious disease. And if you are in one of the vulnerable groups, which are the young, the old, the immunocompromised and pregnant women, this can be a really serious infection for you. And it's mainly associated with ready-to-eat foods. So these are foods that we purchase in the supermarket, we take home, we put them in our refrigerator, and we don't cook them. We eat them directly out of the fridge. So we're talking about things like soft cheeses, and this is an outbreak that occurred in the UK earlier this year, where one person died as a result of consuming this product. We're also talking about things like packaged salads and packaged pre-prepared fruits and vegetables, which is becoming quite a common source of Listeria infections now. And we have been working a little bit on this in recent times. And I suppose this is something that we're noticing when it comes to Listeria, is that it's moving into food groups that we wouldn't have commonly associated with Listeria monocytogenes before. It's cropping up in lots of different places. And also in these plant-based alternatives. So this is from earlier this year, 5 people sick in France related to an outbreak associated with these plant-based dairy alternatives. So this is a real issue for the food sector, for producers of ready-to-eat foods. And I like this quote, actually. I often put this up for my students and ask them to guess the organism. Now, you guys have the inside track already, you know what the organism is, but this is a quote from a vice president of Lando Frost, who produce deli meats, and basically saying here that this organism has changed how the food industry approaches food safety, really. Our scars are numerous and deep, he says. So this is the Irish Listeria monocytogenes Consortium. It's a multi-institute consortium across the island of Ireland, and a number of different universities, and Teagasc leading this consortium. And our remit within this consortium, really, we have had a number of large projects funded through our Department of Agriculture, Food and the Marine, to basically study the behavior of Listeria monocytogenes in foods and to understand it better. So what these projects have facilitated is gathering of a large collection of Listeria monocytogenes strains from the Irish context, sequencing those genomes, looking at them through comparative genomics, looking at their response to stresses in the food processing environment and in the foods themselves, looking at things like biofilm formation, which is really important for Listeria monocytogenes to establish itself in food processing plants, comparing the genome to the phenotype and trying to make connections, genotype-phenotype connections within the collection. Food challenge studies, always a really important thing to do when you're trying to understand the behavior of this organism in foods, because not all foods support the growth of Listeria monocytogenes, and not all isolates will grow in particular foods. Some of them would have a very specific niche. And what I'm here to talk to you about today, obviously, how we use phage in the biocontrol and detection of this organism. And obviously there are commercial live phage products out there on the market that target Listeria monocytogenes. We have ListShield by Intralytics, and we have PhageGuard by Micros Food Safety. And so these are live phage agents. And I suppose one of the issues with these live phage agents is there's no common position on the use of these agents in the food industry across different jurisdictions. So, and I'll come back to talk about that a little bit at the moment. So that's an issue, and that's been an issue for a long time when it comes to using phage products in the food industry. And, there are still hurdles to overcome when it comes to that. In saying that, we still do a lot of work with live phage. We isolate new phages all the time for Listeria monocytogenes. This is one that we published in recently by a previous PhD student of mine, Edel Stone. And the reason that we were particularly interested in this phage P61 is because it's particularly effective against strains of Listeria monocytogenes that grow well on spinach leaves. And there's been quite a lot of talk about Listeria in spinach. In Ireland, we've had several recalls. The Food Safety Authority of Ireland will notify that these recalls are occurring. And what we've also found, which is very interesting, if not a little bit scary, is that the levels of Listeria monocytogenes in spinach can get quite high when the spinach in your fridge looks still very edible. So things like lettuce and things like that, by the time the Listeria gets to a level that will make you ill, it's probably not, you're probably not going to eat it. But with spinach and rocket also, we've discovered that the levels of Listeria can be quite high, and you would still probably eat it directly out of your fridge. But this phage P61 is quite effective against strains of Listeria that grow well in the spinach environment. But what I want to talk to you today about is not the use of live agents, but the use of phage proteins like endolysins, particularly from biocontrol perspective, and also receptor binding proteins of Listeria phages for detection. And because Teagasc, our remit is to support the food industry, what we really want to do is, as well as evaluating these phage-derived proteins from a research perspective, is we want to identify suitable methods to deploy these proteins in the food environment. So we have a large collection of Listeria specific phages, which we've isolated over the years. And I'm going to talk today about this particular one, 293, which was isolated a number of years ago by a former PhD student of mine, Aidan Casey, isolated from mushrooms. We sequenced this phage. It was similar to another one in our collection. So here's 293, very similar to another one in our collection, 188, and similar to other Listeria phages that had been sequenced. We looked to identify the endolysin within this particular phage, and here it is, ORF25. It's an amidase, so it has two domains, a cell wall binding domain and a catalytic domain, which is the endolytic part of the amidase of this particular endolysin. And in previous work that we had done with Staphylococcus aureus phages, we had discovered that if you want to use these endolysins as lysis from without, you actually don't need the cell wall binding domain at all. Sometimes you don't even need to use the whole catalytic domain. So we really focused on looking at the amidase rather than the full endolysin itself. It makes it easier to produce these things recombinantly. And so another former PhD student of mine, Vincenzo Pinone, spent some time in CNB in Madrid with Dr. Mark van Rooij's group, and they recombinantly produced the amidase domain from phage 293. And it's quite a potent anti-listerial agent. You can see here when you add 20 micrograms of the purified amidase domain into the host of the phage 293 itself, you get lysis of the culture in quite a short period of time. You're talking about maybe 10 minutes here to clear the culture. So it has a very potent anti-listerial activity in liquid media, which is all well and good, but how does it fare when you move into a more food processing simulated environment? So we wanted to look at one of the main issues with Listeria monocytogenes in food processing environments is it forms biofilm. And when it forms biofilm, it can be very difficult to remove. And you have a lot of evidence of persistence of this particular pathogen in food processing plants. And its ability to form biofilm is very much a part of that. So here you can see this is the host of phage 293, the strain 473, forming biofilm on stainless steel. And then it's dyed with live dead stain. So the green is live and the red are dead cells. So this is a 4-day biofilm here. If you take the purified amidase 293 and you pre-treat the stainless steel surface with the amidase 293, Listeria monocytogenes or 473 finds it difficult to form biofilm on that surface. If you have a preformed biofilm and you treat that preformed biofilm with the purified amidase, you don't really get a whole lot of change. So there's not a whole lot of difference between this and this. So what we can see is that the amidase protein can inhibit biofilm formation on stainless steel, which is a food contact surface in many food processing plants, but finds it difficult, reasonably enough, I guess, to remove already formed biofilm. And we did a lot of experiments, pilot trials out in the field, out in our pilot processing plants, to try and establish whether these agents could be used as they were purified proteins in the plant. And what we found is that stability is a major issue. So we do get degradation of the protein out on the food contact surfaces and on the non-food contact surfaces, floors, walls, drains, etc., during those pilot trials. So stabilization was a problem. And so we looked to see how we could improve the stability of these particular amidase proteins. And a former colleague of mine who I worked with a number of years ago in the US, Professor Eric Alterman, who at the time was at Ag Research in New Zealand, they had just created these tailored bio-nanoparticles which display any enzyme that you're interested in on the surface. And these are produced by a recombinant E. coli. So what they had been producing at the time was this lytic enzyme from a methanogenic phage, and they were looking to reduce the numbers of methanogens in the rumen. So we set up a similar scheme. So here you go with your Pha-C plasmid. This is producing the bio-nanoparticle. You can clone into this plasmid any enzyme sequence that you like, and the E. coli will produce these beads, which are then displaying the enzyme of interest on the surface. So in our case here, we did this with the full-length endolysin, and we did this with just the amidase domain itself. And here you can see the result of one particular assay. This is in liquid media again, but if you have Listeria monocytogenes here, the blue line is 473, that's the host itself, it's growing away happily. We used the bio-nanoparticles themselves without any enzyme displayed on the surface. You get a little bit of inhibition, but not a whole lot. And then down here, what you have are the bio-nanoparticles displaying either the full-length endolysin, and you get, some inhibition of Listeria, and also with the amidase as well, you get inhibition of Listeria in broth. So we published this last year. So this was very encouraging. This was encouraging that we could use this bead nanoparticle as a stabilization strategy for these enzymes. We have run into some difficulty with producing large quantities of these, but we're still working to to resolve those issues. But we are also looking at other methods to improve the stability of the enzymes and to protect them for delivery in the food processing environment. So we are working with a spin-out company from Teagasc who are experts in microencapsulation. So they are a company that we're working with currently to try and, purify the endolysins and encapsulate them so that we can use them on the surfaces in a food processing environment. One of the other things that we're doing is we're working with Dr. Andrew Flouse, who's a protein biochemist in the University of Galway. And we're working with Andrew to try and develop some sort of plug and play platform for endolysin expression, because what we've learned over the years working with these enzymes is that, if you change to a different endolysin, you're almost reinventing the wheel all over again. So what we want to do is to try and establish a much more stable platform where we can come in and out with different sequences. And his experience using enteins in protein sequences in order to be able to stabilize the proteins is hopefully will help us to do that. So I just want to switch to, for a few minutes, to the work that we're doing using phage-derived proteins as detection probes, and we know that we can use nucleic acids, antibodies, lots of other biological agents as detection probes. We can use the whole phages themselves, and indeed we can use the receptor binding proteins from phages, which are found in the base plate of the phage, as potential detection probes in biosensors. And with this work, I am working with my colleague in Queen's University, who Professor Katrina Campbell, and Katrina has developed over the years a number of different lateral flow devices. We're all very familiar with lateral flow devices these days after a number of years of COVID testing, but basically it's a similar type of a test. And what she had developed prior to us working together was mainly tests for mycotoxins and other biological toxins. So we decided to investigate whether we could use the phage receptor binding proteins from the Listeria phages to develop a similar device to detect Listeria in the food processing plant. So again, we're back to our phage 293, and through a series of mutational experiments and also through some homology searches, you can see here that these open reading frames are found in the base plate. Through homology with other Listeria phages, we identified a number of different receptor binding proteins from numerous phages. I'm just going to talk about this one today. And what we did here was we overexpressed this receptor binding protein. We GFP tagged it so that we could follow it through the system. We purified it. And then we conducted these experiments here. So what we're looking to do is to see, can this receptor binding protein be used for capture of Listeria monocytogenes? And one of the ways that you can see whether there's specificity between the protein that you've produced and the cells that you want to detect is that if you add the protein into the host cell and then you try and infect with the original phage, the protein should block absorption of the phage. So that doesn't happen at low concentrations, but if you increase the concentration to 1.5 milligrams or above, what happens is the protein has bound to the receptors and the phage can no longer bind. If you use a different phage, it doesn't matter because the receptors are different. So this is some evidence that this is specific for this particular Listeria strain, and we can see that it's happening when you use protein above 1.5 milligrams per mil, and it's specific for that particular phage. So we're working to increase our bank of receptor binding proteins, and then we will move forward into biosensor development. So from the point of view of food safety, there are some considerations, I suppose, when using phage enzymes to inhibit bacteria in the food processing environment. They do display potent antibacterial activity. They can inhibit biofilm formation on food contact surfaces. This is really important in the context of the food processing plant. As was alluded to earlier, there's no resistance really being detected to date. We haven't detected it in the lab with any of the endolysins that we've produced or any of the strains that we are working with, and others have reported the same. And it may perhaps be easier to regulate these agents from a food processing point of view than live phage products, which comes with its own issues. The issues, I suppose, with using these enzymes is that they can be expensive and time consuming to produce. As I've demonstrated, the stability in the food processing environment is an issue and is something that we are working to address. And as they are produced natively, these endolysins don't work against gram-negative pathogens because of the outer membrane. But I know others are working on that and have solved those issues in some cases. But there's no common position, and I mentioned this earlier, there's no common position on the use of bacteriophages or their derivatives in food systems. Currently in the EU, they are considered by most member states as food additives. In Ireland, that's certainly the approach that's being taken. So therefore, if you use these agents, the live agents or anything derived from them in your plant, you must put it on the label, and most food producers don't want to do that. It's different in the US. In the US, they are considered as processing aids. So they're like anything else that you'd use in your processing plant, any sanitizer or detergent that you would use for normal cleaning, they don't appear on the label, and the phages are treated just like that. So we've been talking for a long time about how we need to deal with these agents and regulate them, and those discussions are continuing, and I think they're more important than ever now, because these phage preparations could be game-changing for low and middle income countries because they're a cheap alternative to food safety management in these food processing environments. So just some concluding remarks. I suppose to us, this is not a relatively new technology. We've been talking about phages biocontrol agents for a long time. The technology goes back 100 years really, but from a food producer's point of view, what often happens when I talk to food producers about using phage as biocontrol agents is that why would you? A lot of these companies would be dairy producers, so they're trying to eliminate phage out of their plant. They'd be fermented food producers, so they're trying to grow lots of bacteria in their plant and then eliminate phage from the plant as much as possible. So you were talking then about bringing phage in. Obviously, we need to educate our customers in terms of what that actually means, and that it doesn't actually mean that their process is going to be impacted. What we do know from the work that we've done with Listeria monocytogenes and other foodborne pathogens as well is that there isn't a one-size-fits-all solution here. Every matrix that you're dealing with, every food surface that you're dealing with, you need a slightly different approach, and those have to be evaluated in pilot trials. The mode of application is critical, and we've seen that through various studies, and this will be critical to implementation as well. So it might be that application to food surfaces, as opposed to putting phage or phage-derived products on the food itself, may be easier to regulate. And we certainly see that when we survey customers, they don't really want to have viruses or viral products put on their food, but don't mind it so much if it's in the food processing environment. So these are things we have to think about as we regulate these agents. And again, like antibiotics, we're not going to replace antibiotics with phage. It's going to be a complementary approach. This is the same thing here when we're going to use phage in as part of a food safety management strategy. It's going to be part of the hurdle approach. So the different mechanisms that we put in place to prevent pathogens entering the food chain, this will just be another one, and it could be used very much where Listeria is a specific problem. And again, all potential stakeholders here must be engaged in the dialogue regarding the use of these agents, because everybody has something to bring to the table when it comes to discussion around these agents. So I just want to thank all my collaborators here. This is work that's been going on for a number of years. The people in my lab, Aidan, Vincenzo, Idel, and Kieran have done much of the work that you've seen here. I think I've name checked most people on this slide, but the one person that I haven't is Horace Neve, and we worked for many years with Horst, and he's taken a lot of the images that you saw in these slides. Also want to thank our funders, and of course I want to thank you for your attention, and I will take any questions that you have. Thank you very much.